Hello, everyone, and welcome to today's webinar, ICH Stability Testing and Method Development. I'm Tim Wright, editor of Contract Pharma. Our webcast today is being sponsored by Nelson Labs. They're a global provider of microbiological and analytical lab testing services for medtech and pharmaceutical companies. During the broadcast, we're going to learn about stability testing, why it's a vital part of product development and is conducted throughout a product's life cycle. Our speaker is Shiri Hector. She's Nelson Labs' Chemistry Senior Lab Operations Manager. She's an organic chemist and received her master's degree in organic chemistry from Tel Aviv University in 2005. Shiri began her career at Gibraltar Labs in 2006 and started as a chemistry group leader in the analytical chemistry department. After 2 years, she was promoted to the QA department as a quality assurance deputy with a focus on analytical chemistry. During her time in QA, Shiri completed certified quality auditor and certified Six Sigma Green Belt training with the American Society of Quality and currently holds both certifications. In 2017, Shiri moved back into operations as the chemistry department manager, and as the leader of the analytical chemistry department, Shiri oversees all analytical testing and fills the role as the main subject matter expert for analytical testing, giving support to the tech service teams. In today's presentation, she's going to provide a brief overview of the ICH guidelines for stability testing, as well as aspects of analytical method development. We will be accepting questions throughout the presentation. You can send those in using the box at the lower left of your screen, and questions will be answered after in a brief Q&A. And the presentation will be archived on our website, contractpharma.com, for 1 year. And if you're experiencing any technical difficulties, you can type those into the box as well, and someone from the broadcast vendor will help you out. If you're ready, Shiri, I'll turn it over to you.
Okay, I'm ready.
Okay,
hi, everyone. I'm Shiri, and first I want to thank everyone for joining me today. I know those are not easy times, and I do appreciate everyone that could join me today, and I hope you enjoyed the presentation. I'm going to talk about the ICH stability guidelines. I'm going to give a brief overview and also talk about how it correlates to the testing we do at Nelson Labs. So we'll start from the stability definition. And merely stability is how well a product retains its quality over time. And why do we test for stability? We certainly don't want to be surprised when we use the API or the finished product. And we can be surprised because chemical degradation can lead to a change in the concentration of the active ingredient and later lead to a change in the therapy or in the testing we do. And another option could happen that during the chemical degradation, there are going to be toxic products formed, and that's going to affect the end users. So in this slide, I have some examples for potential effects of instability. There can be a loss or increase in the concentration of the active ingredient. And a change in the concentration leads to a change in the therapy. It could be alternation in bioavailability. It could be a change in rate or extent of absorption. An example, if we have aging tablets, will have a change in the dissolution rate. Loss of content uniformity. It could be creaming of emulsions or suspensions, or it can even be precipitation in liquid or injections that we have. Loss of package integrity. A change in the package integrity can cause contamination, and the contamination can be microbial or other contamination to the product. Reduction of the label quality. If the label is compromised, that's going to affect the legibility of the product. So the stability testing objectives is defined in the ICH guidelines. The purpose of the stability testing is to provide an evidence on how the quality of the drug substance or drug products varies with time under the influence of a variety of environmental factors such as temperature, humidity, and light, and to establish a retest period for the drug substance or shelf life for the drug product and the recommended storage conditions. So we can say the scope of the stability testing is to provide the evidence of the quality of the drug, to establish shelf life, to determine the storage conditions, and to determine the container closure system. These are the various types of the stability that exists. When we talk about a drug must possess chemical stability, physical stability, microstability, and therapeutic stability. We can divide the stability studies to the 6 different stages. We have the early stage is stage 1, which are the stressed and accelerated testing on the drug substance, and it goes up to stage 6, which is the follow-up stability studies. So this slide represents the stability guidelines of the ICH. There's 6 sections, Q1A to Q1F. Because we don't have the whole day today, I'm going to mainly focus on Q1A, which is the stability testing of new drug substance and products, and Q1B, which is the photostability testing of new drug substance and products. So in this slide, we can see the ICH stability zones. The zones are divided by the climatic zones, and you can see there's 4 different stability zones. So we have the blue, which is zone 1, the green, which is zone 2, yellow, which is zone 3, and the red, which is zone 4. And here are the climate zones as they're defined in the ICH guidelines. So there's zone 1, which is moderate climate. There's the zone 2, which is the Mediterranean climate. Zone 3 is the hot or dry climate, and zone 4 is very hot and moist. So you can see the average temperatures and the average humidity for each of the climate. So we'll start with Q1A, is the stability testing of new drug substance and drug product. We're going to talk first about the drug substance. And if you can see, there's those are the section in the standard that we're going to talk about. I'm going to, on the next slide, I'm going to explain each of one of them specifically. So the general statement, information on the stability of the drug substance is an integral part of the systematic approach to stability evaluation. Stress testing. So stress testing is similar to the stress test that I'm sure a lot of us go through when we go to a doctor and we get a well checkup. So what we do, we expose the substance or the sample, if it's a drug substance or drug product, we expose it to extreme conditions, and we look for the degradation products. So we often refer to stress testing as forced degradation studies, and we examine the degradation product, and by that, we can establish degradation pathway and developing and validating stability indicating methods. When we say stability indicating method, we mean that the methods are validated and quantitative analytical methods, and those methods can detect a change in the product, and they can accurately measure the assay and impurities. So what should stress testing include? They should include the effect of temperature on the drug, the effect of humidity, oxidation, photostability, and the effect of pH. You can see here those are typical stress conditions. And of course, when we come to choose the stress condition, it's really important that we know what product we're testing or what sample we're testing or drug substance we're testing, because the condition will vary according to the sample we're testing. But those are the typical conditions that work with most of the product and can be a starting point when we do the stress testing. On the next slide, I'm going to go on each one and explain a little more. So we'll start with oxidation. This is one of the most common conducted stress testing. And the selection of the oxidizing agent concentration really depends on the sample in question. We can typically start by placing the drug in a solution of hydrogen peroxide. We can go from the low concentration to 30% hydrogen peroxide at room temperature, and we want to look for a maximum of 20% degradation. pH, the selection of the type of the stress testing, if it's acid hydrolysis or base hydrolysis, will depend on the stability of the sample. Common reagent that we use for hydrolysis, we can use hydrochloric acid from 0.1 to 1 molar for acid hydrolysis, and sodium hydroxide or potassium hydroxide from, again, 0.1 to 1 molar for base hydrolysis. We normally start from room temperature and we see the degradation, and if there's no degradation, we can use higher temperatures. And again, we look for the same 20% degradation. What's important to remember at this step, that the degraded sample that we use needs to be neutralized back after we expose it to acid or base, because we want to avoid further decomposition. We have the thermal stress test, dry heat and wet heat. The solid drug substance and product should be exposed to both thermal stress condition and dry and wet heat, while the liquid drug product should be exposed only to dry heat. The degradation are normally conducted between 40 degrees to 80 degrees. If it's low or high humidity, we can choose based on the product, of course, and the normal, most widely accepted temperature is 70 degrees. It is also recommended to measure the effect of temperature at 10 degrees increment above the routine accelerated testing at humidity of 75 or higher. So how do we select the batches for stability testing? The ICH guidelines give clear instruction on how to select the batches, and this verbiage is actually exactly from the ICH guideline. And in summary, what they want us to do is to select at least 3 primary batches of the drug substance, and they could be manufactured in the minimum pilot scale, but the manufacture procedure should simulate the actual final process of production batches. The quality of the batches of the drug substance that we use for the formal stability studies should be representative for the quality of the material that is made on the production scale. and the container closure system. The stability study should be conducted on the drug substance that is packaged on the container closure system that is the same or simulates the packaging proposed for storage and distribution. The testing frequencies. There's the long-term studies, and the long-term usually go above 2 years. The first year is going to be every 3 months time point testing. The second year will be every 6-month time point testing. And after 2 years, it could be annually. The accelerated storage condition will be minimum 3 points when you include the initial and final points, and it's going to be from a 6-month study. And the intermediate storage conditions would be a minimum of 4 time points, including the initial and final time points from a 12-month study. So the storage condition, this is a general case for storage conditions. The duration here is the duration of the minimum duration requires a time of submission. This is not the overall duration of the stability study. And when we talk about the general case, the storage conditions are chosen by the substance that we need to test. If we conduct long-term stability studies, we can submit data over 12 months stability studies from 3 different batches. But at the time of submission, we need to propose a long enough study that will cover the proposed retest. And if we choose to do a long-term studies on 30 degrees and 65% humidity, then we do not need to do the intermediate studies. Of course, when we do the stability studies, when we do the accelerate alongside with the long-term studies, we need to observe the data and see that there's no significant change occurs at the time of the accelerated storage. And when they talk about significant change, they mean that the product doesn't meet the set criteria or the specification that we set in the beginning of the study. Those are storage conditions for drug substance that are intended to use in the refrigerator or drug substance that are intended to use in the freezer. So the stability commitment and evaluation. So the section about the commitment talks about what I said earlier about the commitment to continue the studies post-approval and firmly establish the retest period. So when the time of submission and we have long-term data only for 12 months, we need to provide a commitment that we're going to continue the stability study and establish the retest period. The evaluation of the stability study, we need to gather all the information we got from the stability study based on the minimum batches of the drug product and use it to establish a retest period for all the future batches that's going to be manufactured under similar circumstances. And when they say the evaluation, it doesn't need to cover only the assay, but also the level of degradation product and the appropriate attributes. The statement and labeling. A storage statement should be established for the labeling. And the statement should be based on the evaluation we did from all the stability data we gathered. And we have to put specific instruction on the label where applicable. So the section 2.2 talks about the drug product. And if you see the categories are similar to what we talked about in the previous slides, and I want to put more emphasis on the photostability testing. So normally the photostability testing is carried out on a single batch of material, and it's always recommended to have a systematic approach when we do photostability testing. So the studies will cover the test on the drug substance, and then test on the drug product outside its immediate pack, test on the drug product in the immediate pack, or test on the drug product in the marketing pack. When we talk about drug substance, photostability should consist force degradations testing and confirmatory testing. So the purpose of the force degradation testing is to evaluate the overall photosensitivity of the material for method development purposes and or degradation pathway elucidation. They designed to provide suitable information to develop and validate test methods that are going to be used later for the confirmatory test. And the reason why they're so important is because when we do the force degradation studies, we're more likely to have degradants that are not going to be formed in the confirmatory test. And that will help us to validate develop and validate the analytical procedure. We can use a variety of exposure conditions depending on the photosensitivity of the substance and the intensity of the light source we use. And the sample should be in chemically inert or transparent containers. The confirmatory testing should be undertaken to provide an information necessary on handling, packaging, and labeling, and they should identify a precautionary measure that needs either in the manufacturing and the formulation, and if we need to use a light-resistant packaging. This is a decision flowchart from the photostability testing for a drug product from the IC8 guidelines. And you can see the starting point and the exposure, and if acceptable change, we can end the testing, but if not, then we have to check the immediate pack. If they're acceptable change, we can end the testing, or we want to change the immediate pack. So photostability testing procedure. There's two light sources options. Option one would be any light source that is designed to produce an output of similar to the D65 or ID65 emission standard. And this is internationally recognized standards for outdoor light. And option two is a combination of both cool white fluorescence and near UV lamp. At Nelson Lamb, we have a photostability chamber that has option two, so we have a combination of cool white fluorescence and near UV lamp. And we use quinine chemical actinometry to actually ensure that the specified light exposure is obtained. So the way we do it, we expose the sample side by side with the validated chemical. The chemical is 2% solution of quinine monohydrochloride dihydrate. You can see it on the bottom of at the right end of the bottom of the slide. And the technique measures the increase in absorbance over time of this chemical versus the same chemical that has not been exposed to the light, which we call it a dark control. And we want to look at the change in absorbance. We're looking for at least 0.5 change in absorbance at 400 nanometers. And a 0.5 change in absorbance will ensure that we expose the sample to the required overall illumination of not less than 1.2 million lux hours and not less than 200 watt-hour per square meter. So it can take from 3 days to 5 days. And if you see, there's an example of how the photostability chamber looks like. At the end of the exposure period, the samples are examined for any physical change, if there's clarity in color or solution, anything that we can see physically. And we also test it for assay and degradant by validated method. So these are type of stability tests that we perform at Nelson Labs. We can perform stability testing for different dosage forms. And every dosage form has different testing that are required. And here we detail all the testing that we can perform. And also on the next slide. A lot of the testing, a lot of the products that we get are finished products, so we don't have a validated method to do the assay and impurities. And when we have a scenario like this, we have to perform method development. The method development will be specifically for a specific formulation. So if there is a change in the formulation, we might need a new method development. We might even use the existing method, but we will have to revalidate the method based on the new formulation. So method development is required when a procedure is not available in the literature. So if we have a new invention, new patent, or new product that we've never seen before, when there's no compendial method available, when the new formulation of the product causes interference with the compendial method, when we want to test lower active concentration, or when the existing analytical procedure is maybe too long and too expensive and we want to improve it. So this is the method development flow. We start with the research. We look at the sample and the active that we need to be the active that needs to be quantified in the sample. And we search in the literature or in the compendia any references for something similar that we can use as a start point. We look at the formulation information to understand what excipients there are in the product that can interfere with the testing. We look at the chemical structure and possible side reaction that could occur in the middle of the testing. And we gather all this information to choose the appropriate method. So when we talk about stability indicating method, the method needs to be, as I said before, specific and quantitative. So methods like chromatography method like HPLC, GC, LCMS, GCMS, or even UV or IR methods are good, but when we have titration methods, those are not stability indicating method because titration methods, they're not specific and they're not able to identify the degradant. So even if the titration method will show you a reduction in the concentration over time during the stability study, it will not be able to identify the degradants. And a lot of times, those degradants can cause interference in the actual titration method. So titration is usually a not good method for stability indicating method. After we choose the appropriate method, we will go to the experiments. Oh, sorry. So the method development testing usually includes a feasibility test. So we'll try to take a common method from the research we did And we make some required modification according to the sample we have. Usually we start with the sample prep. And then we take the method and we optimize the parameters for better results. So if we do chromatography, we want to make sure the peak shape is okay, there's no tailing, we optimize the instrument conditions. Recovery studies, this is a very useful tool to sample the spike with a known amount of the active and see how much we can recover. And it's very useful when we deal with related substances or impurities. We perform limit of detection, so we want to know what the detection limit of the method and limit of quantification. And we perform force degradation studies. After the method development is completed, we make a conclusion to see if this is the method we developed is suitable for validation, or we need to maybe try a new approach because the recovery is low, or we couldn't get the peak that we wanted. And if the method is suitable for validation, we go and fully validate it based on the ICH guidelines for method validation. So this kind of ends up my presentation for today, and we're going to move to the Q&A.
Yeah, that's great, Sherry. Thanks so much for that presentation. It's really informative. We've got a bunch of questions here, so I'm just going to jump right in. First one asks here, are stability requirements different for generic IV drugs?
Okay, so that's a very good question. I would want to say yes, but actually they're not really different. I mean, they can be slightly they're slightly changed for the fact that you can submit for less data points, but you still need to make a commitment for the full stability study, and what you can do is use a drug master file for the drug substances in your finished product. The FDA actually has a guidance on stability for generic drugs, so I can send it if required.
Okay, great, thank you. Let's see here. What are the conditions for validation of specificity, and what are acceptable conditions for stressing the product?
So I think for stressing the product, we talked about it in the presentation. That was slide 16. There's a typical condition for stress testing.
Okay.
And in regards to specificity, when you do the specificity, you want to make sure that there's no interference in the diluents that you're using in the method, or there's no interference of the degradant. So the method should be specific. So if, example, we talk about chromatography, there is no interference in the retention time of the active ingredient.
Okay.
And the specificity usually done by running a diluent and checking to see if there's any, and when running the sample and see that nothing interferes in quantifying your active.
Okay, very good. Let's see here, question 4. I'm going to push this slide back to the questions here. How does stability information in the lab correlate to the actual stability of the formulation?
Well, when you do the stability study, you want to know this information because when you actually formulate, it's important to know the drug substance stability because that can affect your formulation. If you know the drug substance is not stable in specific condition, when you go to the formulation, you don't want to use these conditions. So.
Okay. And how many lots are recommended to be pulled for stability studies?
They recommend 3 batches.
3 batches?
Mm-hmm.
Okay. And how do you predict the shelf life of a product drug using the accelerated stability studies?
That's a very good questions, and there is a way to do that mathematically. You can take the Arrhenius equation and calculate from that what would be equivalent to a 1 year at a certain temperature. So if we take 20 degrees, let's say, temperature, we can calculate mathematically what would be equivalent to 365 days at 20 degrees. So an example, 40 degrees, if we choose 40 degrees accelerated conditions, the number of days you need would be 90 days at 40 degrees, and that will be equivalent to a 1 year at a long-term stability study or real-time stability studies.
Okay, great. Let's see here. How do you choose temperature and RH set points?
So, like I said in the presentation, we choose the temperature and humidity condition based on the sample that we actually want to test. We need to know the sample. If the sample breaks down at a higher temperature, then we're not going to choose higher temperature. And we also need to look at the climate zones to choose the condition.
Okay, thanks. Let's see what's next here. How will regulatory agencies react to mistesting pull points due to recent events?
That's a very good question. I'm not sure I have an answer because these recent events, I think it's something new to everyone, even the regulatory agencies. I don't think anybody ever experienced a pandemic like this before.
I guess we're all learning as we go.
Yeah. But I think what the regulatory want to see is they want to see justification and risk assessment to justify why the missed testing, not to justify why you missed the testing, to justify why it will be okay if we missed those time points. And that's going to be based on the risk assessment.
Okay. Let's see here, moving on. This person would like to know if the ICH guidelines are for pharma only, or can they be used for personal care products as well?
The ICH guidelines talk about drug substance and drug product, but there are guidelines for personal care and cosmetics. There's an ISO guideline, and I think that's going to be more applicable for the personal care product.
Okay. And whoever asked that question, Shiri, can follow up afterwards and provide more detail. Next question here asks, in general, how long does it take to develop a stability indicating degradation impurity, such as 2 known impurity test method?
We normally take 4 to 6 weeks to develop a method from scratch, doing also the degradation studies. With that said, we first do, as I said, the research. So the research probably takes the longest time to have a set-up approach to know how we're going to test the sample, and then we do the experiments.
Okay, great. Let's see here. What are the stability testing requirements for IND-enabling studies?
What are the stability? Oh, okay. So for which is the investigational new drug application. The information, I guess, vary. It depends on the phase of the investigation or the proposed duration of the investigation and the dosage form. So there's also specific guidelines on that, and I can send it separately.
Okay, perfect. Let's see, we have time for a couple more here. What are the biggest challenges facing your industry given the current COVID-19 crisis?
Okay, so my biggest challenge is senior lab operation manager and as a central employee that come into work every day, and my lab employees come into work every day. So the biggest challenge is really to keep up with the workflow without risking anyone's health and practice safe methods for or social distancing. Trying to mitigate the risk, we tend to not have more than specific people at the same time at the lab, so we have to do shifts. And really, it's a big challenge for us to get used to the new schedule and also keep up with the demand, because the demand is still the same. People, regarding the COVID-19 crisis, people are still unfortunately sick from other things. People are still need drugs. So the demand is still there, but we're working on very limited conditions. Another challenge is that all of the other companies are working in limited conditions, so there's a longer lag time to receive solvents or reagents that we need for the testing. So a lot of times it will make the testing longer.
Just curious, have you, with people, customers you've spoken with, are there any kind of projections that you're looking towards for how long, the disruption will continue?
I think so far at our area they said until mid-May, and they will review it again. So it changes between state to state.
Yeah.
We're like that until mid-May. But another challenge is that even if they go back to normal, it's not going to be maybe normal in the beginning. So we're still going to see some challenges. I have to say that everyone is in the same boat, and everybody is really understanding and understand the delay, and that's very nice.
Yep. Okay. Let's see. I think we got time for one more here. What is the standard deviation accepted by agency if the concentration of DRU is 1 ppm concentration in aqueous medium? I don't know if I...
I think they meant concentration of the drug is 1 ppm. If the drug is 1 ppm, and you're talking about an assay and not impurity, I would say that you still have to go through, they would maximum go to 5% RSD, so the method needs to be robust. But if you're talking about impurities can be 10 or even 15% RSD.
Okay, perfect. All right, so I think we're going to wrap up the Q&A here. I'd like to thank Shiri again for your great presentation, very informative, and thanks again to our sponsor, Nelson Labs. If you would like more information, this slide that's up on your screen will guide you in the right direction. And we did have a bunch more questions come in, and Shiri and the folks over at Nelson will have those in hand and be able to follow up with you directly after the fact. And as I mentioned earlier, this session is going to be archived on our website for 1 year, contractpharma.com. So thanks very much, everybody, for joining us, and have a great rest of your day.